All right, friends, before we get into this one, I need you to do something for me. Picture this, you're floating, somehow 50 kilometers above the surface of Venus. Down below you, the ground is hot enough to melt lead. The pressure down there would crush a submarine. But up where you are, in this one specific layer of cloud, the temperature is basically room temperature, and the pressure is basically Earth normal. And drifting past your face, in tiny concentrations, is a gas that on our planet comes from exactly one place, 99% of the time: the metabolic waste of microbes living without oxygen. That gas is phosphine, and in September of 2020, a team led by astronomer Jane Greaves announced they'd found it in the clouds of Venus. And for about a week, half the planet lost its mind a little bit. Because, think about it, Venus. The planet we basically wrote off decades ago as a lava-soaked, acid-rained hellscape. And suddenly, there's this one molecule that doesn't really have a good explanation that isn't, on some level. Something is alive up there. So that's where we're starting today. But honestly, that's just the opening act. Because friends, today is one of those long ones. The kind where I've basically been hoarding rabbit holes for weeks. And now, I'm just gonna dump all of them on you in one sitting. Because it turns out they're all connected by the same underlying question, which is, what if the universe, and even our own planet, is way weirder than what we got taught, and the only reason we haven't noticed is we've been looking with the wrong tools, in the wrong places, assuming the wrong definition of normal. We're gonna talk about life that might be hiding on Earth right now, using a totally different chemistry than anything in your biology textbook. We're gonna talk about how a bird's eyeball and a leaf might both be running on quantum physics, and what that could mean for how we one day build spacecraft. We're gonna get into time crystals, and whether the future can reach back and tap the past on the shoulder. And we're gonna end out in the cold, dark space between stars, where some genuinely strange chemistry has been quietly cooking up the building blocks of life for billions of years, possibly including the ones that ended up becoming you. So yeah, get a drink, get comfortable. This is a long one, and honestly it's one of my favorite kinds of episode to make, because almost none of this is settled science. A lot of it is the stuff actual researchers are arguing about right now, this year, in actual journals. Let's get into it. So let's start close to home. Like as close to home as it gets Earth, right now today. There's a concept in biology and astrobiology called the shadow biosphere. And the basic pitch is this. What if there's a whole category of microbial life on this planet that we have simply never detected, not because it's rare or hiding in some inaccessible trench, but because our entire detection apparatus is built to find one specific kind of life, and this other stuff just doesn't show up on the test. Here's the thing you have to understand about how we look for microbes. Almost every method we use, sequencing ribosomal RNA, running things through a PCR machine, growing samples in a Petri dish with nutrient broth, all of it assumes the organism uses DNA or RNA, builds proteins out of the same 20-ish amino acids every textbook lists, and generally plays by the biochemical rules that all known life on Earth happens to share. And here's the kicker, the thing that should bug you a little bit once you hear it. Every single organism we have ever identified, from bacteria to blue whales, shares that same basic biochemical toolkit. Not similar toolkits, the same one, which on one hand makes total sense if everything descended from one common ancestor, like the textbooks say. But it also means that if there were a second, totally independent origin of life sitting right next to ours, using, say, a mirror image version of our molecules, or a different set of amino acids, or some exotic backbone instead of DNA. Our microscopes and our gene sequencers wouldn't flag it as biology at all. It would just look like background noise, or get tossed out as a sequencing error, or never get sampled in the first place because nobody thought to look there. This idea got formalized in the mid-2000s, mainly by a philosopher of science named Carol Cleland, working with Shelley Copley, and they actually coined the term shadow biosphere for it. The idea got a lot more attention a few years later, in 2009, when Cleland teamed up with physicist Paul Davies and a handful of other astrobiologists. Stephen Benner, Christopher McKay, Charles Lineweaver, Felisa Wolf-Simon, and published a paper basically laying out what we should even be looking for if we wanted to go hunting for this stuff. Things like organisms with the wrong chirality, meaning molecules that are mirror images of the normal versions. Organisms using a different genetic code than the one every known life form on Earth shares. Or biochemistry built around a different backbone element entirely. And friends, here's where I have to pause and ask you something, because I think about this a lot. If you were a scientist convinced there might be a hidden form of life sitting in plain sight, basically invisible to the entire field's existing toolkit, where would you even start looking? Because the obvious answer is look at things we already can't fully explain. And it turns out there's a real candidate for exactly that. Cleland has specifically pointed at something called desert varnish, that dark, almost black coating you sometimes see on rocks in deserts, the stuff ancient peoples used to scratch petroglyphs into, because the dark surface contrasts so nicely with the lighter rock underneath. Scientists have argued about whether desert varnish is even biological or purely a mineral process since basically the time of Darwin. Nobody's fully settled it. Cleland's argument is, look, this is a textbook case of exactly the kind of overlooked anomaly a shadow biosphere candidate should look like. Something weird, ambiguous, sitting in plain sight on every continent, that we've mostly just shrugged at for over a century instead of properly investigating with the could this be a second kind of life question in mind. And beyond just pointing at one weird rock coating, the 2009 paper actually laid out a broader practical strategy for this kind of hunt. And I think it's worth walking through, because it's a genuinely clever piece of scientific detective work. Their basic recommendation was to cast a wide net, rather than trying to directly culture or sequence a hypothetical shadow organism, which assumes you already know roughly what you're looking for. You instead screen environmental samples for chemical anomalies that don't fit the known biological pattern, but also don't look like ordinary, explainable geology. Things like an unexpected skew in the ratio of left-handed versus right-handed versions of a molecule in a sample that should be chemically neutral on that front, or an isotope ratio of carbon or nitrogen that doesn't match the signature any known terrestrial organism leaves behind, or some polymer-like structure that's clearly not random mineral buildup, but also doesn't match any known protein or nucleic acid. The pitch is basically, don't go looking for life directly, go looking for anomalies that are hard to explain any other way, and then investigate those anomalies specifically with the shadow biosphere hypothesis in mind, rather than assuming their measurement error and moving on, which, let's be honest, is probably what's happened to plenty of genuinely interesting anomalies over the decades of normal lab work, just quietly filed away as noise. Now, I do have to give you the cautionary tale here, because it's a really good one and it's directly relevant. Felisa Wolf-Simon, one of the co-authors on that 2009 Shadow Biosphere paper, made huge headlines in 2010 for a different but related claim that a bacterium called GFAJ1, found in Mono Lake in California, could substitute arsenic for phosphorus in its DNA backbone. Phosphorus is one of those elements every biology class tells you is absolutely essential, one of the non-negotiable ingredients of life as we know it. NASA held a press conference. It was a huge deal. And then, over the following couple of years, multiple independent labs tried to replicate the result and basically couldn't. The bacterium turned out to just be really good at scavenging trace phosphorus, even in arsenic-rich conditions, not actually swapping it into its DNA. The claim didn't hold up. And honestly, friends, I think that story is worth sitting with for a second, because it cuts both ways. On one hand, it's a real demonstration of science working the way it's supposed to: bold claim, public scrutiny, replication attempts, correction. That's the system functioning correctly, even if it's embarrassing for the people involved. But on the other hand, you can kind of see why a working scientist might think twice before publicly chasing weird life claims after watching that play out. There's real reputational risk in being the person who said, "I think I found something nobody else can see." Especially in a field already filled with people quietly suspicious that astrobiology spends too much time speculating and not enough time confirming. So if you ask me, and you didn't, but I'm gonna tell you anyway, I think the shadow biosphere hypothesis survives that controversy just fine, because the underlying logic of it doesn't depend on any one bacterium being special. It depends on a much more boring, much harder to argue with point. Our detection methods have a blind spot by design, and a blind spot doesn't announce itself. That's kind of the whole nature of a blind spot. And there's a genuinely fascinating, very current twist on that exact chirality idea that I have to mention, because it flips the whole shadow biosphere concept on its head. Instead of asking whether mirror chirality life might already exist naturally, hidden, undetected, a serious chunk of the scientific community spent 2024 and 2025 worrying about whether humans might accidentally build it on purpose. Synthetic biologists have gotten genuinely good at manufacturing mirror image versions of biological molecules, proteins, sugars, nucleic acids built from the opposite-handed chemical building blocks compared to everything in nature. And some labs had started exploring whether you could go all the way and assemble a complete, self-replicating mirror bacterium out of nothing but those reversed molecules. In December of 2024, a group of 38 scientists, including multiple Nobel laureates, published a commentary in the journal Science, specifically arguing against pursuing that goal, accompanied by a nearly 300-page technical report. Their core worry is this: a mirror bacterium would behave totally normally in isolation, growing under the same physical conditions as ordinary bacteria. But its reversed molecular shape means none of the natural immune defenses, predators, or viruses that keep ordinary bacterial populations in check would actually recognize it as a threat, the molecular lock and key. That lets our immune systems and natural ecosystems fight off pathogens simply wouldn't fit. So a mirror bacterium that managed to escape containment could, in theory, spread largely unchecked through ecosystems that have no evolved defense against a threat shaped like that, since nothing in nature has ever had to deal with one before. Now, to be fair to the science here, this remains genuinely contested even among experts. Some synthetic biologists argue a mirror organism would actually be at a steep competitive disadvantage in the real world and likely wouldn't survive well outside a lab at all. And full mirror bacteria don't currently exist and are estimated to be at least a decade away from being technically achievable, if they're achievable at all. But several of the actual researchers who'd been pursuing this line of work, including a synthetic biologist named Kate Anomaly, read through the risk analysis and voluntarily stopped their own research as a result. I bring this up specifically because it's such a strange, mirror image echo, pun fully intended, of the shadow biosphere idea we started this episode with. The same exact chirality criterion that astrobiologists use to ask, could undetected weird life already be hiding on Earth, is also, right now, at the center of a live, serious debate about whether humans might accidentally manufacture a version of exactly that scenario ourselves. Okay, so here's where it gets bigger, because this idea doesn't stay confined to Earth. Paul Davies has this argument he calls something like a cosmic imperative, the idea being, if life really did arise independently more than once on a single planet, right here, under our feet, then that's actually a pretty strong hint that life isn't some impossibly rare cosmic fluke. It's the kind of thing chemistry just sort of tends to do, given the right conditions, given enough time. And if that's true, it should be happening elsewhere in the solar system too, not just on far-off exoplanets we can barely image, but on places we can actually go visit with a spacecraft. It's basically a more modern, more biochemically specific cousin of the famous Drake equation, that back-of-the-envelope formula astronomer Frank Drake came up with in 1961 to try to estimate how many communicating civilizations might exist in our galaxy. By multiplying together a chain of factors like the rate of star formation, the fraction of stars with planets, and the fraction of those planets where life, then intelligence, then detectable technology actually shows up. The Drake equation never really gives you one solid number, because half its terms are still basically educated guesses, but it's useful precisely because it breaks an impossibly huge question down into a chain of smaller, more answerable ones. And the shadow biosphere idea slots right into one of the earliest, most foundational links in that chain. It's a direct, testable way of asking, how easily does life get started, given the right conditions, using data from right here on Earth instead of waiting on a signal from across the galaxy? Which brings us right back to that Venus Phosphine story I opened with. So, quick fact check on myself here, because I want to be straight with you about where this stands. The 2020 detection was real, multiple telescopes picked it up, and the team specifically tried to rule out known non-biological sources like lightning, volcanic outgassing, or meteor impacts, because the concentration they found was way higher than those processes are thought to produce. And this is a real, but follow-up analysis in 2022, some of it from members of the original team, raised doubts about the original signal, suggesting it might have been partly an artifact of sulfur dioxide contamination in the data. So as of right now, this is genuinely unresolved. It's not confirmed life. It's not debunked. Nonsense either. It's sitting in this frustrating scientific limbo where the only real fix is more data, which is actually happening. There's a mission concept called VERV that would hitch a ride on the European Space Agency's Envision mission, specifically to map biosignature gases like phosphine and ammonia in the Venusian clouds. And NASA's also got DAVINCI and VERITAS in the pipeline for Venus. So within roughly the next decade, we might actually know. And look, friends, I'll be honest with you about the part that I personally find the funniest slash most humbling part of this whole thing. Even if we do confirm there's something biological going on in the clouds of Venus, those organisms would still have to be tolerating an atmosphere that is almost entirely sulfuric acid. Like on Earth, the most acid-tolerant microbes we know of can survive maybe 5% acidity in their environment. The clouds of Venus are not 5% acid. They are basically just acid. So if life is up there, it's not just alien in the sense of not from Earth. It's alien in the sense of operating under chemical conditions that would dissolve basically anything we know how to keep alive in a lab. Wild to think about. Mars gets the same treatment, just underground instead of in the clouds. There's been this long-running, deeply frustrating mystery around methane on Mars. NASA's Curiosity rover carries an instrument called SAM, short for Sample Analysis at Mars, which has been sniffing the Martian air on and off since 2012. And starting around 2014, researchers began noticing the background methane level seemed to rise and fall with the Martian seasons, peaking in local summer. On Earth, methane behaving like that is overwhelmingly a biological signature, produced either by living microbes or by the geological breakdown of ancient biological material. But other careful statistical reanalyses of that same rover data, run by independent teams, have failed to confirm a strong, consistent seasonal cycle. Once you account for all the ways trace gases can leak in and out of an instrument sitting on a moving rover. So right now, the methane story on Mars is also stuck in that same maddening, interesting, but not resolved, zone. The working theory, if there is something alive down there, is that it would be living in the subsurface, in pockets of liquid water kept from freezing by salt content or geothermal heat, getting its energy not from sunlight, but from chemical reactions with the surrounding rock. What's called chemosynthesis. The same basic strategy some extremophile microbes use in Earth's deep subsurface and around hydrothermal vents. And then there's the ocean moons, which honestly might be the strongest case of all four targets, just because the conditions check more boxes simultaneously. Europa, one of Jupiter's moons, has a subsurface ocean that's estimated to hold roughly twice the volume of water in every single ocean on Earth combined, sealed under an icy shell. NASA's Europa Clipper mission is out there right now specifically to study that ice shell and look for places where ocean water might occasionally reach the surface, because any organic material riding up in that exchange would be a lot easier to detect than trying to drill miles down through solid ice. And then there's Enceladus, a much smaller moon of Saturn, which doesn't even make you wait for an exchange. It's got geysers. Actual plumes of water. Ice and vapor erupting straight out of cracks in its surface, shooting material directly into space, where the Cassini spacecraft was able to fly through the plume and sample it directly. And what Cassini found in there wasn't just water, it found amino acid precursors. And in a 2018 analysis, actual large, complex organic macromolecules, heavier and more structurally elaborate than anything found on comets. Then, in 2023, researchers went back through Cassini's old archived data one more time and found something that genuinely surprised a lot of geochemists. Phosphates, meaning the moon's ocean actually contains phosphorus, in concentrations the team estimated at roughly 100 times higher than in Earth's own oceans. That one matters a lot, because phosphorus is one of the 6 core elements every biology textbook lists as essential for life as we know it, right alongside carbon, hydrogen, nitrogen, oxygen, and sulfur. And up until that point, nobody had confirmed it existed in an ocean anywhere beyond Earth. So as of that discovery, Enceladus became the only other place in the solar system where we've directly confirmed every single one of those 6 essential bioelements is present in liquid water. None of that is proof of life. To be really clear about that, complex organic chemistry, even with every essential element present and accounted for, is not the same thing as biology, even though it's a necessary stepping stone toward it. But it does mean Enceladus has basically done half our sampling work for us, free of charge, just by spraying its insides into the void where a passing spacecraft could catch some. And honestly, friends, I can't talk about hunting for biosignatures in our own solar system without bringing up the absolute soap opera that's been unfolding around a planet way outside it, because it's basically the exact same story playing out on a bigger stage, in real time, while I've been writing this episode. There's an exoplanet called K218b, sitting about 124 light-years away, orbiting a small red dwarf star. It's bigger than Earth, and the leading theory is that it might be what astronomers call a Hycean world, a planet with a global ocean underneath a thick, hydrogen-rich atmosphere. In 2023, and then again with stronger data in April of 2025, a team using the James Webb Space Telescope reported picking up hints of a molecule called dimethyl sulfide, or DMS, in that atmosphere. And DMS matters specifically because on Earth, it's produced almost exclusively by marine life, mainly phytoplankton. It's actually part of why the ocean smells like the ocean. So a tentative DMS signal on a faraway ocean world is about as close as you can get to astronomy waving a little biology flag at you. And then, predictably, exactly what happened with the Venus phosphine story happened again here. Multiple independent teams went back and reanalyzed the same James Webb data using different statistical methods, and the result has been genuinely chaotic, in the best scientific sense of that word. Some reanalyses couldn't replicate the DMS signal at all, and instead found the spectral features were just as easily explained by far more boring molecules like ethane. Other reanalyses confirmed methane and carbon dioxide just fine, but found no statistically significant evidence for DMS specifically. A NASA-led study using fresh modeling found multiple non-biological, purely photochemical pathways that could plausibly produce DMS-like signals without life being involved at all. And one particularly blunt paper, published in late 2025, is literally titled, and I'm not exaggerating the bluntness here, K218b does not meet the standards of evidence for life. So as it stands right now, heading into the back half of this decade, the honest scientific consensus on K218b is that the original exciting claim has not held up well under scrutiny, even though the planet itself remains a genuinely fascinating target and nobody's ruling out future observations, changing the picture again. I bring all that up because I think it's actually the perfect closing note for this whole segment of the episode. Friends, Venus, Mars, the ice moons, and now K218b are all, every single one of them, running through the exact same cycle. Exciting tentative signal, huge headlines, careful reanalysis, walked back confidence, more data needed. And if you're the kind of person who finds that frustrating, I get it, I really do. But I gently push back on the frustration a little, because that cycle isn't a sign that astrobiology is a sloppy field full of false alarms. It's actually the opposite. It's what a genuinely difficult, high-stakes question looks like when serious people refuse to declare victory prematurely just because the first headline was exciting. Finding life elsewhere would be, arguably, the single biggest discovery in the history of our species. And the fact that scientists keep dragging every promising signal back through the ringer before accepting it is, frankly, exactly how it should work. So here's my honest take, if you want it, on all four of these: Venus, Mars, Europa, Enceladus. I don't think any single one of them is a slam dunk. I think if you force me to bet on which one we confirm first, if any, I'd probably put my money on one of the ice moons, just because the sampling logistics are friendlier. You don't have to drill. You don't have to survive sulfuric acid. You just have to fly through a plume that's already erupting on its own. But what I find genuinely moving about this whole shadow biosphere framework, the thing that keeps me coming back to it, is that it forces this slightly uncomfortable question on us. What if the reason we haven't found alien life isn't that it's rare, but that we keep bringing the wrong measuring stick? What if it's been staring back at us this whole time, in a desert varnish stain, in a cloud layer we wrote off 20 years ago, and we just don't have the right kind of eyes yet to see it as what it is? All right, let's shift gears completely, because this next one is one of those topics where, the first time I read about it, I had to put my phone down and just stare at a wall for a minute. Friends, I want you to look at a leaf. Doesn't have to be a real one right now, just picture one. Green, sitting in the sun, doing the most basic, most taken-for-granted biological process there is: photosynthesis. Light goes in, energy comes out. Everybody learned the chlorophyll diagram in 9th grade. Boring, right? Except here's the thing nobody tells you in 9th grade. There's a real, ongoing scientific case that the way that energy actually moves through the molecular machinery inside that leaf might be relying on quantum coherence. The same spooky, hard-to-pin-down quantum weirdness that physicists usually need to chill down to near absolute zero inside an isolated lab apparatus just to even observe it for a few microseconds. And it might be happening at room temperature, inside a leaf, while a bug is probably crawling on it. And honestly, friends, this whole idea is way older than you'd think. It's not some brand new internet science fad. Back in 1944, the physicist Erwin Schrödinger, the same guy whose name is attached to that famous cat thought experiment, gave a series of lectures that got turned into a short, hugely influential book called What is Life? In it, he speculated that the stability of genetic material might rely on some kind of quantum mechanical effect, since ordinary classical physics struggled to explain how something as small and seemingly fragile as a gene could stay so remarkably stable across generations. He didn't have anything like modern molecular biology to work with. DNA structure wasn't even worked out until almost a decade later. But that book is famously cited by both James Watson and Francis Crick as one of the things that pulled them toward studying genetics in the first place. So this conversation about whether quantum mechanics is secretly load-bearing inside biology isn't a recent curiosity. It's basically been simmering at the edge of biology and physics since before we even knew what DNA looked like. Here's the actual science behind that claim, because I don't want to just throw quantum leaf at you and walk away. There's a structure called the Fenner-Matthews-Olsen complex, found in certain photosynthetic bacteria, whose entire job is to grab photons of light and funnel that energy super efficiently toward a reaction center where it actually gets used. Back in 2007, a research team led by Greg Engel used a technique called two-dimensional electronic spectroscopy, basically a way of hitting a sample with precisely timed laser pulses, and reading out an extremely detailed signal, and found oscillating patterns in that signal that looked a lot like the signature of quantum coherence, meaning the energy wasn't just bouncing around randomly from molecule to molecule like a pinball. It was behaving more like a wave exploring multiple paths simultaneously before choosing the most efficient one. A follow-up study a few years later found similar signals even at physiological body temperature-ish conditions, which was the real shocker, because the conventional wisdom up to that point was that quantum coherence is way too fragile to survive in anything warm and wet and chemically chaotic, like, a living cell. Now, I have to be straight with you here, because this is exactly the kind of claim where the popular science version runs way ahead of where the actual physicists are. There's real, ongoing disagreement in the field about how to interpret those oscillating signals. Some researchers argue they really do represent genuine quantum coherence doing useful work in photosynthesis. Other researchers argue the oscillations might instead be explained by something called vibronic coupling, essentially classical vibrations in the molecular structure that can produce similar-looking signals without needing true quantum coherence to explain the efficiency. So if somebody tells you it's proven that plants are quantum computers, that's a bit ahead of the actual data. What's fair to say is, there is a real, published, ongoing scientific debate about whether quantum effects are doing meaningful work inside one of the most fundamental biological processes on the planet. And that on its own is a pretty remarkable place for biology and physics to be colliding. Okay, here's a question for you, and I genuinely want you to sit with it for a second before I tell you the next part. If a plant might be quietly exploiting quantum mechanics just to harvest sunlight a little more efficiently, what else in biology might be doing something similar, hiding in plain sight, that we just haven't thought to test for yet? Well, friends, turns out there's a really strong second example, and this one's got my personal vote for the single weirdest confirmed-ish phenomenon in this whole field: bird navigation. Specifically, European robins, and how they figure out which way is north when they migrate. For a long time, the leading guess was something boringly mechanical, like tiny magnetite particles in the beak acting like an internal compass needle. Turns out that's probably not the main story. The leading theory now is something called the radical pair mechanism, and it lives inside a light-sensitive protein in the bird's retina called cryptochrome, specifically a version called Cry4. Here's roughly how it's supposed to work. Light hits the cryptochrome molecule and kicks out a pair of electrons that end up with correlated, or you could even say entangled, spin states. Earth's magnetic field then subtly influences how those two electron spins evolve over time relative to each other, which in turn changes the chemical products that come out the other end of the reaction. And because that chemistry is happening across the curved surface of the bird's eye, the pattern of chemical output essentially maps onto the direction the bird is facing relative to the magnetic field. So the bird might genuinely be seeing the magnetic field, almost like an overlay on its normal vision. And the experimental support for this isn't just theoretical hand-waving. Researchers have done these incredibly clever experiments where they expose robins to the normal, static geomagnetic field, plus a second, weak oscillating radiofrequency field, tuned to match the specific quantum mechanical frequency you'd expect from that radical pair mechanism. When they pointed that oscillating field perpendicular to the Earth's magnetic field, the birds got disoriented and basically lost their sense of direction. When they pointed it parallel instead, the bird's navigation snapped right back to normal. That's a really specific, really hard-to-fake signature. It lines up exactly with what you'd predict if quantum spin dynamics are doing the actual work, and it's genuinely difficult to explain. With a purely classical, magnetite compass-style model, there's even a deeper theoretical puzzle buried in here that I think is honestly the coolest part. For the radical pair mechanism to work as a sensitive compass, those two electron spins need to stay coherent, meaning stay correlated with each other in that delicate quantum way, for long enough that Earth's comparatively weak magnetic field actually has time to nudge the outcome. But a bird's eyeball is warm, wet, and absolutely packed with random molecular jostling, which is exactly the kind of environment that's supposed to destroy quantum coherence almost instantly. So how does it survive long enough to matter? One proposed answer from a physicist named Ioannis Kaminis is that a different quantum effect called the quantum Zeno effect. Basically, the idea that frequent enough measurement of a quantum system can actually freeze its evolution and protect it from decay might be naturally stabilizing the coherence inside the radical pair, almost like the surrounding biological noise is accidentally protecting the very quantum effect you'd expect it to destroy. If that's right, it would mean evolution somehow stumbled onto a way to exploit one of the more counterintuitive corners of quantum mechanics, just to help a small bird figure out which way is south for the winter. I don't know about you, but I find that almost funnier than it is impressive. And before you ask, yeah, friends, there's real, serious engineering interest in this. If we can fully understand how a biological system manages to use quantum spin dynamics for direction finding under warm, messy, real-world conditions, that's directly relevant to anyone trying to build practical quantum sensors that don't need to be cooled down near absolute zero in a giant dilution refrigerator just to function. There's active research into bio-inspired quantum sensing and navigation systems, partly motivated exactly by trying to reverse engineer whatever trick the bird's eye is pulling off. So this isn't just a fun fact about robins, it's a live engineering target. And since we're on the subject of senses doing unexpectedly quantum things, I have to bring up the genuinely strangest, most contested member of this whole family: smell. There's a long-running, decidedly minority scientific theory, most associated with a biophysicist named Luca Turin, which proposes that your nose might not actually identify odor molecules purely by their shape, the way the mainstream lock and key model of olfaction assumes, where a molecule fits into a receptor sort of like a specific key fitting a specific lock. The competing idea is that olfactory receptors might instead be sensing the molecular vibrations of an odor molecule through a process involving electrons tunneling across the receptor and losing a tiny, specific packet of energy that matches the molecule's particular vibrational frequency, essentially smelling the molecule's internal hum rather than its outline. It's a genuinely clever idea, and it would explain a few odd experimental quirks the shape-only model struggles with, like certain molecules with totally different shapes still smelling weirdly similar to each other. But friends, I want to be really clear that this one sits on much shakier ground than the photosynthesis or bird navigation stories. It remains a minority position. Several experiments designed to directly test it have come back ambiguous or unsupportive, and most working olfaction researchers still favor the conventional shape-based explanation. treating the vibrational tunneling idea as an intriguing but unproven outlier rather than an accepted part of how smell works. I include it here specifically as the cautionary example in this segment, proof that not every quantum mechanics in biology claim is created equal, and the photosynthesis and magnetoreception stories earned their stronger standing through years of converging, replicated, increasingly specific evidence that this one, so far, just hasn't managed to match. Now I do want to walk back one thing that gets repeated a lot online, because the user-generated version of this topic tends to overreach in a specific direction, and I'd rather correct it than let it slide. You'll sometimes see claims that tardigrades, those tiny, almost indestructible eight-legged microorganisms that can survive being frozen, dried out, boiled, and blasted with radiation, survive radiation specifically because of some quantum tunneling trick. As far as the actual published research goes, that's not really what's going on, at least not as the primary mechanism. What the actual biochemistry shows is a protein called D-sup, short for damage suppressor, discovered by a University of Tokyo team in 2016 in a tardigrade species called Rhizodius variornatus. D-sup is a disordered, sprawling protein that physically binds to the tardigrade's DNA, where it's wound around structures called nucleosomes, and it basically acts like packing foam, shielding the DNA from the hydroxyl radicals that radiation tends to generate, which are the actual molecules that go around shredding genetic material. It's a totally classical, structural biochemistry mechanism, really elegant, genuinely Nobel-worthy levels of clever in its own right, but not quantum mechanics. So if someone hits you with tardigrades survive radiation because of quantum tunneling, you can politely tell them that's mixing up two genuinely cool but separate stories. That said, and this is the honest caveat I promised you at the top of this episode, quantum tunneling absolutely is a real, well-documented phenomenon elsewhere in biochemistry, just not in tardigrade radiation resistance specifically. Hydrogen and proton tunneling have been shown to play a measurable role in certain enzyme-catalyzed reactions, letting chemical transformations happen faster than you'd predict if you only allowed particles to behave classically, basically letting protons cheat through an energy barrier rather than going over it. Biochemist Judith Klinman and her collaborators spent years building the experimental case for this, mainly by studying how reaction rates change when you swap a normal hydrogen atom for one of its heavier isotopes, deuterium, in certain enzyme. Reactions, the size of that change, what's called the kinetic isotope effect, ends up way larger than ordinary classical chemistry predicts, and tunneling is the explanation that actually fits the numbers. That body of work is now pretty well accepted as a real, functioning feature of how certain enzymes speed up chemistry inside your own cells, every single day, whether you're aware of it or not. There's also a long-standing, more speculative line of research into whether proton tunneling between DNA base pairs could occasionally contribute to spontaneous mutations. That one's been theorized since the mid-20th century and is still actively studied, but it remains a more contested, harder to pin down claim than the enzyme tunneling work. So I'd file that specific one under plausible and interesting, not settled. And actually, since we're already on the subject of tardigrades, friends, I want to give them their proper, non-quantum mechanical due, because the real story is honestly impressive enough without inventing a quantum angle for it. Back in September of 2007, The European Space Agency strapped a payload of dehydrated tardigrades to the outside of a capsule called Phosphine and sent them into low Earth orbit for about 10 days in an experiment with the not at all subtle name TARDIS. Some of the tardigrades were shielded from solar ultraviolet light, but exposed to the open vacuum of space and full cosmic radiation. Others got the full, unfiltered treatment: vacuum plus cosmic radiation plus direct solar UV, the harshest combination they could throw at them. When the capsule came back down and researchers rehydrated the samples, the vacuum and cosmic radiation group came back to life in huge numbers. And even a chunk of the group that took the full solar UV blast on top of everything else survived, woke up, and went on to lay viable eggs that hatched into normal offspring. That made tardigrades the first animal ever documented surviving that exact brutal combination of conditions, a result published in Current Biology in 2008. And it's still the foundational data point everyone cites when they talk about tardigrade resilience in actual space conditions, not just in a lab freezer. NASA's been studying the gene expression side of that resilience on the International Space Station, more recently too, trying to figure out exactly which genes the animal switches on under spaceflight. Stress. So no, it's not quantum tunneling letting them shrug off cosmic radiation. It's an extraordinary, very classical biochemical toolkit, DSUP included, that happens to be good enough to survive conditions that would kill basically anything else we know of. And honestly, I think that's a better story anyway, because it doesn't need a hand wave toward quantum mysticism to be remarkable. Sometimes the universe doesn't need to be quantum to be wild. Sometimes regular biochemistry, refined over hundreds of millions of years, gets you all the way to survived the vacuum of space and had kids about it. So zoom out with me for a second on this whole segment, because here's where I land on it personally. And again, you didn't ask, but you're getting my take anyway. I think the photosynthesis coherence story and the bird magnetoreception story are genuinely two of the strongest, most experimentally grounded cases we have anywhere in biology for quantum mechanics doing real, functional work inside a living organism, not just existing at the level of, well, atoms are quantum, technically. And I think the fact that both of those examples involve something as mundane as plant leaves and migratory birds, not exotic lab-grown bacteria, not extremophiles in a volcanic vent, just regular organisms doing regular things in your backyard, is what makes this whole field feel less like a fringe curiosity and more like a genuinely underappreciated layer of how biology actually works. There might be a whole undiscovered category of quantum biology tricks sitting in plain sight in totally ordinary organisms, the same way the shadow biosphere idea suggests there might be a whole undiscovered category of life sitting in plain sight in totally ordinary places. Same blind spot, different scale. Okay, friends, new segment. And this is the one where I'm gonna ask you to really buckle in, because we're about to get into time. And time is one of those topics where it's incredibly easy to make things sound way more mystical than they actually are. So I'm gonna try really hard to walk the line here between this is genuinely mind-bending, and this does not mean what your group chat thinks it means. Let's start with something a contractor would never, ever do. Imagine building the walls and the roof of a house, and only afterward pouring the foundation underneath it, and somehow the house still stands up just fine, like the order never mattered at all. That's roughly the vibe of what we're about to talk about except instead of a house, it's the universe, and instead of a contractor, it's quantum mechanics. Let's do time crystals first, because even though the name sounds like something out of a fantasy novel, it's a real, published, Nobel laureate-originated idea. And there's actual hardware in labs right now demonstrating versions of it. Back in 2012, physicist Frank Wilczek, who'd already won a Nobel Prize for completely unrelated work, was teaching a class about ordinary crystals, the kind you already know about, where atoms arrange themselves into a repeating pattern in space, like the carbon lattice inside a diamond. And he had this thought: if matter can spontaneously organize into a repeating pattern in space, why couldn't it organize into a repeating pattern in time instead? Picture a structure that, even sitting completely still in its lowest energy state, would cycle through sequence of configurations forever, returning to where it started at regular intervals, with literally no energy input required to keep the cycle going. Like a clock that needs no battery and never winds down. Now here's the part where real physics did its job and corrected the original idea pretty quickly. Other physicists pointed out that Wilczek's original version, a time crystal sitting in its actual lowest energy ground state, would basically amount to a perpetual motion machine. And perpetual motion machines violate the laws of thermodynamics. Full stop. No exceptions. That's about as settled as physics gets. By around 2014 and 2015, separate groups had produced what's called a no-go proof, essentially a mathematical argument that Wilczek's original ground state version simply can't exist. So friends, this is a great example of science self-correcting in real time, and also a great example of how the most interesting part of a story is sometimes what happens after the first cool idea gets shot down. Because physicists didn't just drop the whole concept, they pivoted. Instead of looking for a time crystal in a system's resting ground state, they started looking at driven systems, things you periodically kick or pulse with energy from the outside, and asked whether you could get a stable pattern that repeats at some multiple of that driving rhythm, rather than at the same rate you're poking it. That version is called a Floquet time crystal, named after a piece of 19th century math originally developed to describe periodically driven systems generally. And it's a genuinely strange result once you sit with it. You're tapping the system at one steady rate, say once every second, and instead of just bobbing along in step with your tapping, the system settles into a rhythm of, say, once every 2 seconds, locking onto a submultiple of your driving frequency, and refusing to budge from it even if you nudge the timing or strength of your taps around a bit. That stubborn, self-protecting refusal to just follow the beat you're giving it is exactly the kind of spontaneously organized behavior that earns it the word crystal. And unlike Wilczek's original concept, it doesn't violate thermodynamics because you're continuously feeding energy in from outside the system. And this version has actually been built. In 2021, a team working with Google's Sycamore quantum processor, the same chip Google used a couple years earlier to make headlines about quantum computers, solving a calculation that would have taken a classical supercomputer 10,000 years, used 20 qubits to demonstrate what's called a prothermal time crystal, where ions repeatedly flip between two states in a pattern that survives for a meaningfully long time before the whole system eventually settles down into boring thermal equilibrium. The way everything in physics eventually does, if you wait long enough. And it kept getting better. In 2025, a team at the University of Colorado Boulder managed to build something genuinely new. A time crystal you can actually see with your own eyes, no quantum computer required. They used rod-shaped liquid crystal molecules, the same general category of material that's inside your phone screen, sandwiched between two die-coated glass plates. And under the right lighting conditions, those molecules start swirling into colorful, looping, never-resolving patterns that you can literally watch happen under a microscope, persisting for hours. And separately, also in 2025, a Finnish research team went a different direction and managed to couple a continuous time crystal to an actual physical mechanical oscillator, getting the two systems to lock into sync with each other, with the time crystal's rhythm staying coherent for minutes at temperatures near absolute zero, which, if it holds up and scales, opens the door to time crystals as functional components inside future quantum sensors or ultra-precise on-chip clocks, not just lab curiosities. And friends, this isn't purely an academic curiosity sitting in a physics journal somewhere either. There's real, practical interest building around this stuff. Because a time crystal is, by definition, a system that holds a stable, self-sustaining rhythm without needing continuous outside intervention to keep that rhythm precise. Researchers have started floating it as a potential building block for next-generation timekeeping, the kind of ultra-stable on-chip clock you'd want inside a quantum sensor or a precision navigation system. The Finnish team's 2025 result, where they got a continuous time crystal to lock into sync with a physical mechanical oscillator and stay coherent for minutes at a stretch, is specifically being talked about as a step toward hybrid chips that could plug a time crystal's rhythm straight into existing microwave or photonic hardware. And the visible, liquid crystal version out of Colorado Boulder has people speculating about much weirder, lower-tech applications too, like anti-counterfeiting patterns for currency, on the logic that a pattern which is provably, perpetually never repeating in the same way, is a genuinely hard thing to fake. None of that is built and shipped technology yet, to be clear. We're talking about applications still being sketched out on whiteboards and in early lab prototypes. But it's a good reminder that weird new phase of matter, and might end up in your phone in 15 years, aren't actually mutually exclusive categories, historically speaking. But here's the part I really need you to hear clearly, because this is exactly where the popular imagination runs way past the actual physics. None of this means time travel. None of it. A time crystal is not a wormhole. It's not a device for revisiting yesterday. It doesn't let information move backward through time. What it actually is, and I promise this is still genuinely remarkable even with the time travel stuff stripped away, is a brand new phase of matter, the same conceptual category as solid, liquid, or gas, except this one is defined by its behavior in time rather than its arrangement in space. That's the whole claim, and honestly, discovering an entirely new category of how matter can organize itself is already a huge deal on its own. It just doesn't need the time travel framing to be impressive, and adding that framing actually undersells what's true about it in favor of something that isn't. All right, now let's get into the part of this segment that's genuinely contested, even among people who do this for a living: retrocausality. The pop science framing usually goes something like, quantum physics proves the future can change the past. And the example almost always cited is something called Wheeler's delayed choice experiment, or its more elaborate cousin, the delayed choice quantum eraser. The basic setup involves sending individual particles through a kind of double-slit arrangement, where depending on how you choose to measure them afterward, you either get evidence the particle behaved like a wave passing through both slits at once, or evidence it behaved like a single particle going through just one. The genuinely strange part is that in some versions of this experiment, you can delay your choice of which measurement to make until after the particle has already, in some physical sense, already passed through the apparatus and supposedly already decided how to behave. Which sure sounds like your later decision reached backward and changed an earlier event. So here's my question for you, friends, and I want you to actually think about it before I tell you where the physics community has mostly landed on this. If a choice you make later genuinely could rewrite what already happened earlier, would you even be able to tell the difference between that and just not yet understanding the full picture of what was actually happening the whole time? Because that second option is basically what most careful analyses of these experiments conclude. There's a really thorough paper by David Ellerman from 2015 that walks through exactly why the delayed choice experiments don't actually require backward-in-time causation to explain. And the short version of the argument is something he calls the separation fallacy. The mistake of assuming the particle had already fully, definitely decided to be a wave or a particle at the earlier moment, when actually it remained in a sewer position, an undecided, both-at-once state, the entire time, right up until the measurement that finally resolves it. Once you stop assuming an earlier definite outcome existed to be retroactively changed, the whole paradox kind of evaporates. There's no past being rewritten, because there was no fixed fact in the past to begin with. The later measurement isn't reaching backward in time. It's just the first moment any definite fact actually came into existence at all. And honestly, most working physicists who have looked closely at this agree the popular, retrocausal, framing oversells what's actually going on. And I promise this, but is the genuinely interesting part. This isn't a fully unanimous, case-closed situation either, which is exactly why I wanted to include it in this episode, rather than skip it for being already debunked. There are real, published, minority interpretations of quantum mechanics that do formally build retrocausal elements into their mathematics, on purpose, as actual physics rather than mysticism. John Cramer's transactional interpretation, developed back in the 1980s, describes quantum events as a kind of handshake between a wave traveling forward in time from the source and a second wave traveling backward in time from the eventual detector, the two of them meeting partway to produce what we experience as a completed quantum event. There's also something called the two-state vector formalism, developed by Yakir Aharonov and collaborators, where a quantum system gets described using two separate quantum states simultaneously, one evolving normally forward from the past, and one evolving backward from a future measurement, with the present somehow sandwiched between both. And this one isn't purely theoretical either. It's actually led to real, hands-on laboratory experiments. Researchers have used a related technique called weak measurement, which lets you extract a small amount of information about a quantum system without fully collapsing it the way a normal measurement would, to do things like reconstruct the average trajectories photons seem to take as they pass through a double-slit setup, building up a picture that looks remarkably like classical, continuous paths in experiments published in mainstream physics journals back in 2011. So unlike, say, a purely armchair philosophical argument, The two-state vector formalism has genuinely been used to make testable predictions that hold up in real apparatus, even though its broader retrocausal interpretation remains a minority position. Both of these are mathematically rigorous, peer-reviewed frameworks, and crucially, both are constructed specifically so that they reproduce all the same experimental predictions as standard quantum mechanics, meaning even their own proponents argue these models don't let you actually send a signal backward in time. You couldn't use them to, say, text your past self a winning lottery number. They're reinterpretations of the underlying machinery of reality, not loopholes you could exploit. Now, where this gets genuinely speculative, and I want to flag clearly that we're now solidly in theoretical physicists having fun at a whiteboard territory, rather than anything close to confirmed science. Some researchers have explored whether retrocausal models might help untangle some of the trickiest open puzzles in physics, like certain fine-tuning problems in cosmology, or some of the deep weirdness around information and black holes, or conditions right around the very early universe. And since I brought up black holes specifically, let me actually ground that one in the mainstream version of the puzzle for a second, because it's worth knowing the real story doesn't need retrocausality at all to already be strange. Back in the 1970s, Stephen Hawking showed that black holes slowly radiate particles and eventually evaporate away completely. And his original calculation suggested that whatever detailed information fell into the black hole over its lifetime would simply be destroyed in the process, gone, with only a generic, featureless glow of radiation left behind. That's a real problem, because quantum mechanics has a hard rule that information is never supposed to be truly destroyed, just scrambled and hidden, in principle always recoverable if you could track every particle closely enough. For decades, that contradiction sat there as a genuinely unsolved paradox, with serious physicists on opposite sides of a famous bet about it. Then, starting around 2019, several research groups working with tools borrowed from string theory found a way to calculate the problem more carefully, using something called quantum extremal surfaces. And the new math strongly suggests information actually does gradually leak back out in the Hawking radiation as the black hole evaporates, fully consistent with ordinary forward-in-time quantum mechanics, no backward causation required. So even one of the most genuinely paradox-feeling problems in modern physics has, in the last several years, been trending toward a resolution that keeps time pointed firmly in one direction, which I think is actually a nice, grounding note to end this part on, because it's a reminder that deeply weird and counterintuitive, and requires throwing out causality, are not the same category, even though they get conflated constantly in pop science coverage. I want to be really direct with you about where the retrocausal versions stand by comparison. There is currently no experimental evidence supporting genuine backward-in-time information flow at the scale of black holes or cosmology, and most of that specific framing remains squarely in the realm of theoretical exploration, rather than anything close to an accepted explanation. If you ever see a headline that flatly states, "Scientists prove the future affects the past," what's almost certainly underneath that headline is one of these contested, minority interpretation papers, presented with way more confidence than the actual physics community would give it. And here's the thing I want to leave you with on this whole segment, because I think it's the part that actually matters most. Time crystals and retrocausality get lumped together a lot in casual conversation, probably because they're both weird time stuff. But they are not the same phenomenon, and they're not even really related to each other beyond both challenging our gut intuitions about how time is supposed to behave. A time crystal is about a structure repeating itself rhythmically through time, the same way a crystal lattice repeats itself spatially. It doesn't involve information moving backward at all, just forward, on a loop. Retrocausality, in the versions where it's seriously discussed, is about whether later events can have any kind of formal causal influence on earlier ones within the existing mathematics of quantum theory, not about loops or repetition. If you take away just one thing from this whole segment, friends, let it be this. Physics is absolutely capable of producing results that sound exactly like science fiction premises, and you do not need to inflate them into actual science fiction premises for them to be worth your attention. The real version is already strange enough. All right, last big topic, and I actually think this one might be the most quietly mind-blowing of the whole episode, even though it doesn't come with anything as flashy a name as time crystal. Friends, picture the recipe for life. The actual molecular ingredients, sugars, amino acids, the basic organic chemistry that eventually got assembled into the first living cells, being slowly cooked up in a kitchen colder than anywhere on Earth has ever been. Light years from any star, possibly before our entire solar system had even finished forming. That's basically the premise of what astrochemists call dark chemistry. And it's one of those research areas that's so new, and frankly, so undercovered outside of academic journals, that I think most people have genuinely never heard of it. Now, I do need to clear up something right at the start here, because the way this topic sometimes gets framed online is, strictly speaking, not physically possible. And I promised you at the top of this episode that we weren't going to fudge facts just to make a cooler story. You'll sometimes see this framed as chemistry before the Big Bang. And I have to be honest with you, that phrase doesn't really mean anything under our current understanding of physics. Chemistry requires atoms. Atoms require protons, neutrons, and electrons to come together in a stable way. And that didn't happen until roughly 380,000 years after the Big Bang, in an event called recombination, when the universe finally cooled enough for electrons to settle into orbit around nuclei instead of zipping around as a free, superhot plasma. Before that point, you didn't have atoms. You had a soup of particles way too energetic to bond into anything. So there's no chemistry before the Big Bang under any framework physics currently has. What's actually fascinating, and what I think this idea was probably gesturing toward, is the chemistry that got going almost immediately after, and especially the genuinely strange chemistry that's still happening right now, today, in the freezing dark clouds scattered between stars. So let's actually go there, because the real story doesn't need any embellishment. The very first molecule to ever exist in the universe is thought to have been something called helium hydride, written as He+, just a helium atom and a hydrogen nucleus stuck together. The simplest possible molecule you could build out of the very first elements that existed. For decades, this was purely theoretical, predicted, but never actually observed anywhere in space, until 2019, when a team using NASA's SOFIA Airborne Telescope, basically an infrared observatory mounted in a modified Boeing aircraft that flies above most of the atmosphere's interference, finally detected helium hydride's signature in a planetary nebula a few thousand light-years away. So that's a genuinely first of its kind discovery, confirming a piece of very early universe chemistry that had been predicted on paper for decades, before anyone actually caught it happening. But honestly, friends, the first molecule of the universe is really just the opening line of a much longer, much weirder story. And that story is mostly set inside what astronomers call molecular clouds. These enormous, dense, incredibly cold pockets of gas and dust scattered throughout galaxies, sitting at temperatures around 10 degrees above absolute zero, which is colder than anywhere that's ever existed naturally on Earth. You'd think a place that cold and that empty would basically be chemically dead, just inert gas drifting around doing nothing. It's the opposite. These clouds are some of the most chemically productive environments we know of anywhere in the universe. Using radio telescopes, facilities like the IRAM 30-meter telescope, the Nobyama Observatory, and increasingly, ALMA, the giant array of dishes in Chile, astronomers have now detected well over 200 distinct molecules drifting around inside these clouds, picked up by the unique radio frequency fingerprint each molecule's rotating, vibrating structure gives off. And it's not just simple stuff like hydrogen and carbon monoxide. We're talking about glycolaldehyde, which is the simplest possible sugar molecule, first confirmed in a molecular cloud in the mid-2000s, and then specifically detected in 2012, using ALMA in the disk of gas and dust swirling around a young, sun-like protostar, meaning this sugar was present in basically the same kind of environment our own solar system would have formed out of billions of years ago. We're talking about urea, ethanolamine, a whole alphabet soup of what chemists call complex organic molecules, generally meaning anything built from 6 or more atoms, forming on the icy surfaces of tiny dust grains floating in these clouds, catalyzed by reactions that wouldn't necessarily even work the same way in the open vacuum of space. The dust grain acts almost like a tiny cold workbench where atoms can land, stick around just long enough to bump into each other, and react. A huge amount of this molecular hunting happens to center on one specific, almost absurdly productive patch of sky called Sagittarius B2, a giant, dense, star-forming molecular cloud sitting near the very center of our galaxy, practically in the shadow of the supermassive black hole at the Milky Way's core. Sagittarius B2 has turned out to be such a ridiculously rich chemical environment that it's basically become astrochemistry's favorite test subject. Researchers have run decade after decade of spectral line surveys across it using telescopes like the IRAM 30-meter dish, specifically because it keeps turning up new molecules nobody had previously detected anywhere in space. It was inside Sagittarius B2, back in the early 2000s, that astronomers first reported tentative radio signals, matching both the simplest sugar, glycolaldehyde, and more controversially, the simplest amino acid, glycine, which is exactly the claim that kicked off the messy back and forth I'm about to walk you through. And then there's glycine, the simplest amino acid, which has had honestly one of the messiest, most back and forth detection histories in this entire field. A team claimed a tentative detection of interstellar glycine back in 2003. A different team published a pretty thorough rebuttal just 2 years later in 2005, arguing the signal didn't hold up to closer scrutiny once you accounted for spectral confusion from other molecules in the same frequency range. As of right now, a confirmed, broadly accepted detection of glycine specifically inside a molecular cloud remains elusive, genuinely contested, which I think is actually a useful, honest example of how messy and iterative this kind of cutting-edge spectroscopy really is, rather than the clean, confident, "we found it" story you usually get in a headline. But here's where it gets really interesting, because while glycine in the open interstellar medium remains shaky, glycine itself has been confirmed, repeatedly, somewhere else, inside actual physical samples we've gotten our hands on. The Murchison meteorite is a great example to actually picture, friends, rather than just take my word for as an abstract data point. It fell on a Sunday morning in September of 1969, breaking apart in the atmosphere over the small town of Murchison in Victoria, Australia, scattering fragments across paddocks and roads, while a fireball lit up the sky bright enough that witnesses compared it to a flare. Locals gathered up more than 100 kilograms of the stuff in the days afterward, some of it reportedly still warm, and because it landed in a well-populated farming area rather than somewhere remote, scientists got unusually fresh, well-preserved samples to work with almost immediately, before rain or contamination had much chance to mess with the chemistry. That meteorite has gone on to become one of the most intensively studied rocks in the history of science, precisely because of what was locked inside it. More than 80 distinct amino acids, identified across decades of increasingly sensitive lab analysis. And crucially, the isotope ratios and the mix of left versus right-handed molecular forms strongly suggest those amino acids, or at least their direct chemical precursors, really did form somewhere out in space rather than after the rock landed on Earth. And in 2009, scientists analyzing material that NASA's Stardust spacecraft had physically scooped up from the tail of Comet Wild 2 and flown back to Earth reported finding glycine in that sample too. So even with the open cloud detections still being argued over, we've got direct physical evidence, sitting in actual labs, scooped out of an actual comet and an actual meteorite, that these molecules really do form out there. And here's a fun, very recent connective thread, friends, that ties this whole segment right back to the K2-18b story from earlier in the episode. That same molecule, dimethyl sulfide, the one at the center of all that exoplanet drama, has itself now turned up in the interstellar medium, identified in 2025 through careful radio spectroscopy of molecular cloud material. So the exact compound everyone was arguing about as a potential alien ocean biosignature is also, independently, just one more entry on the long list of organic molecules the universe seems to be perfectly happy manufacturing out of cold dust and starlight, no biology required. Which doesn't settle the K2-18b debate one way or the other, but it does underline exactly the point those skeptical reanalyses kept making. DMS has real, demonstrated, non-biological ways of forming, so finding it somewhere is nowhere near the same thing as finding life there. There's also a newer, more theoretical thread of research, trying to explain why these molecular clouds keep generating more and more chemical complexity, instead of just topping out at a few simple molecules and stopping. A 2022 study published in the Proceedings of the National Academy of Sciences modeled the chemistry of these clouds as an interacting network, rather than a list of isolated reactions, and found that complexity tends to emerge as a natural consequence of enough simple molecular building blocks interacting with each other over long enough timescales. Basically, given a large enough chemical alphabet and enough time, the cloud's chemistry naturally starts writing longer and longer words. without needing some special extra ingredient to push it toward complexity. That's a pretty elegant explanation for why we keep finding bigger, more elaborate organic molecules out there the more carefully we look. And it dovetails really nicely with the cosmic imperative idea Paul Davies was making back in the Shadow Biosphere segment. Given the right raw materials and enough time, complexity might just be where chemistry naturally tends to drift, on its own, without anyone or anything pushing it there on purpose. Which brings us to the big philosophical payoff of this whole segment, the idea, sometimes called molecular panspermia. Not the more dramatic, often overhyped version of panspermia you might have heard about, where fully formed living microbes supposedly hitch a ride between planets. This is the much more modest, much better evidenced version, which just proposes that the basic building blocks of life, the amino acids and sugars and similar molecules, might have originally formed out in the interstellar medium, then gotten delivered to the early Earth via asteroids and comets, especially during a violent period called the Late Heavy Bombardment, somewhere between roughly 3.8 and 4.1 billion years ago, when our young planet was getting absolutely pummeled by debris left over from the solar system's formation. Under this idea, those impacts weren't purely destructive events. They may have also functioned as a delivery service, seeding early Earth's chemistry with a head start it didn't have to build entirely from scratch. And just to be thorough, friends, I do want to flag the much weirder, much more speculative cousin of this idea, mostly so you can correctly identify it if you ever run into it. Back in 1973, none other than Francis Crick, the same Crick who co-discovered the structure of DNA, teamed up with chemist Leslie Orgel to publish a paper proposing something called directed panspermia. The idea that life on Earth might have been deliberately seeded by an earlier, technologically advanced civilization somewhere else, sending microorganisms out on purpose, rather than life just hitching a ride accidentally. I want to be completely clear that this was always presented by its own authors as a speculative, fairly fringe thought experiment, explicitly intended to provoke discussion rather than as a serious claim backed by evidence, and it has essentially no observational support behind it even 50 years later. I only bring it up because it's a genuinely fun historical footnote, and because it's such a clean example of the difference between credentialed scientist publishing a deliberately speculative idea to spark debate, and credentialed scientist making an evidence-backed claim. Those are two very different categories of statement, even when they come from the exact same famous person. I do want to give you the honest caveat here too, because this is exactly the kind of place where it's tempting to round. The ingredients of life form easily in space. Up to life itself is basically guaranteed to form everywhere. And that's a genuinely big, unjustified leap. There's an enormous, still poorly understood gap between, here are some amino acids and sugars floating around, and here is a self-replicating, metabolizing, evolving organism. Plenty of researchers who study the actual origin of life on Earth would tell you that gap, sometimes called the transition from chemistry to biology, remains one of the single hardest open problems in all of science, way harder than just assembling the raw ingredients. There are competing, actively researched hypotheses about how that final leap might have actually happened here on Earth. One popular framework, often called the RNA world hypothesis, proposes that self-copying RNA molecules came first, and only later got wrapped in membranes, and paired up with proteins to become something we'd recognize as a proper cell. while other researchers favor scenarios centered on the mineral-rich, chemically energetic environment around deep-sea hydrothermal vents as the more plausible cradle. Neither camp has a fully worked-out, experimentally confirmed answer yet, and that's sort of the whole point. Having the ingredients sitting around, even in abundance, still leaves the genuinely hard part of the story largely unsolved. So friends, if you want my honest opinion on this one, and again, you're getting it whether you wanted it or not, I think the interstellar chemistry story is rock solid and genuinely beautiful on its own terms. The universe is, almost as a byproduct of ordinary star formation, manufacturing the basic vocabulary of biology over and over again, everywhere, for free, on a staggering scale. But I think it's important to hold that fact separately from any claim about how likely life itself is to actually show up once those ingredients land somewhere hospitable. Those are two different questions. And right now, we have a pretty good, increasingly confident answer to the first one, and basically no solid answer at all to the second. Okay, friends, let's bring this whole thing back together, because we covered a lot of ground today. And I think there's something genuinely worth pulling out of all four of these stories, once you look at them side by side, instead of as four separate, unrelated rabbit holes. We started on Earth, with the idea that there could be an entire shadow biosphere of weird, undetected life hiding in plain sight, simply because our scientific tools were built to recognize one specific kind of biochemistry and nothing else. We zoomed out to Venus, Mars, Europa, Enceladus, and even a faraway exoplanet, and found the exact same basic story playing out on a planetary and interstellar scale. Tantalizing signals that don't fit easily into a definitely not biology box, but that also, every single time, so far have failed to clear the much higher bar of definitely is. Then we went small, down into individual cells and the eyeballs of migratory birds, and found a totally different kind of hidden in plain sight story. Actual, functioning quantum mechanics, the same physics that needs a giant cryogenic refrigerator to demonstrate in a lab, potentially happening for free, at room temperature, inside completely ordinary living things, doing useful biological work that evolution apparently stumbled onto by accident. Then we went into the deep end of theoretical physics, where matter that repeats itself forever through time has actually been demonstrated in working hardware, while the flashier, more headline-friendly idea that the future can reach backward and rewrite the past turned out to be mostly a misunderstanding of what was happening in those famous delayed choice experiments, with only a small, genuinely contested minority of physicists keeping a formal, mathematically rigorous version of backward-in-time causation alive in their models. And then we ended out in the freezing dark between the stars, watching the universe casually assemble the basic vocabulary of biology, sugars, amino acid precursors, the works, as an almost incidental byproduct of ordinary star formation, happening over and over again, everywhere, for free, on a scale that's honestly hard to wrap your head around. And if you string all four of those together, here's the thread I keep coming back to. In basically every single case, the universe turned out to be more chemically generous, more physically strange, and more quietly inventive than the simplest version of the story we tend to default to. Life's basic ingredients aren't some impossibly rare cosmic jackpot. They're apparently a routine output of stars being born. Quantum mechanics isn't confined to physics labs running at a fraction of a degree above absolute zero. It might be quietly doing load-bearing work inside a bird's eye on a normal Tuesday afternoon. Matter isn't limited to the 3 or 4 phases you learned about in school. There's an entire new category defined by behavior in time rather than position in space. And we've got actual hardware demonstrating it. And the line between definitely just chemistry and could plausibly be biology turns out to be a genuinely difficult, contested, constantly being relitigated boundary. Not some clean, obvious threshold you can spot from across the room. I also think there's a really important, slightly less flashy lesson buried in how often these stories follow the exact same emotional arc: huge, exciting announcement, immediate skepticism from other researchers, careful reanalysis, and usually some amount of walking the original claim back, at least partially. Venus Phosphine did it. K2-18b's DMS did it twice. The arsenic bacteria story did it hard. And I think if you're not used to watching science happen in close to real time, that pattern can feel like a letdown, like the universe keeps almost giving us something amazing, and then snatching it away at the last second. But I'd really encourage you to flip that framing around, because that exact cycle, bold claim, public scrutiny, replication attempts, correction or confirmation, is not a bug in how science works. It is, pretty much definitionally, how science is supposed to work, especially around claims this consequential. If we ever do get a confirmed detection of life beyond Earth, I genuinely hope it goes through exactly this same ringer, gets picked apart by skeptical colleagues from a dozen different angles, and survives all of it anyway. That's what would actually make me believe it, way more than a single exciting press conference ever could. And honestly, friends, I think we're sitting in a genuinely good moment to be paying attention to all of this, because there's real, concrete stuff coming down the pipeline in the next several years that could meaningfully move several of these stories forward at once. Europa Clipper is already out there, on its way to study that ice shell up close. The Verve mission concept, if it gets the green light to ride along with the European Space Agency's Envision spacecraft, would specifically target exactly the phosphine question at Venus that's been left hanging since 2020. NASA's DAVINCI and VERITAS missions are both still in the Venus pipeline as well. James Webb keeps collecting more data on K2-18b and other temperate subneptunes, so that debate is far from finished. And radio telescope. Arrays like ALMA keep doing deeper, more sensitive surveys of molecular clouds, which means the list of confirmed interstellar molecules, including, maybe, finally, a solid, unambiguous detection of interstellar glycine, is probably going to keep growing. Further out, there's an even bigger swing coming, at least on paper. NASA has a flagship concept in early development called the Habitable Worlds Observatory, specifically designed from the ground up to directly image Earth-sized rocky planets around nearby sun-like stars and read their atmospheres for actual biosignature gases like oxygen, methane, and ozone. The same general idea behind the K2-18b debate, but built around hardware purpose-made for exactly that question instead of a telescope, like James Webb, that was repurposed toward it after the fact. NASA's been running community workshops and selecting industry proposals to mature the technology, with early test demonstrations targeted for this year. I do want to be straight with you that this one comes with a genuinely big asterisk attached, though it's currently projected for a launch sometime in the 2040s, carries an estimated price tag north of $11 billion. And its funding has already been a bit of a political football, with one recent federal budget proposal floating a dramatic cut to its near-term funding compared to what Congress had previously approved. So put a mental pin in it as a real, serious, technically grounded plan rather than vague hand-waving about future technology. But also don't expect it to settle anything within this decade. None of this guarantees we get a clean answer to any of the big questions in this episode within, say, the next 10 years. But it does mean these aren't just academic curiosities gathering dust in old journal articles. They're active, moving, well-funded research fronts, with new data landing on a pretty regular basis, which is exactly why I find it more fun to revisit topics like these every so often rather than treating any one episode as the final word. If I had to leave you with one closing thought, friends, it'd be this. I think the honest, humble version of the universe is full of wonders. Isn't actually. We've already found a bunch of cool aliens and quantum tricks and time-bending physics. Because mostly, we haven't, not with the kind of confirmed certainty that would make a textbook. The honest version is closer to. We keep finding signals, anomalies, and theoretical possibilities that are tantalizingly close to something extraordinary. And the actual extraordinary thing about all of it is how much careful, patient, frequently disappointing work real scientists are willing to put in, chasing leads that mostly don't pan out, on the chance that one of them eventually will. That's a less flashy story than scientists discover. Aliens, sure. But I happen to think it's a better one because it's actually true, and because it means the door on every single one of these mysteries is still wide open, right now, for any of you watching who might end up being the person who finally walks through it. So friends, here's what I'd actually love to know from you, and I genuinely mean this. I'm not just saying it to pad out the video. Out of the 4 big things we covered today, the shadow biosphere hiding possibly right here on Earth, the quantum tricks plants and birds might be pulling off without even knowing it, the time crystals and the whole retrocausality mess, or the dark chemistry quietly assembling the ingredients of life out in the freezing dark between the stars. Which one actually got you? Which one made you put your phone down for a second, or go, "Wait, what?" out loud to an empty room? Drop it in the comments, because I read basically all of them, and it genuinely helps me figure out which of these rabbit holes to go deeper on next time, since honestly I could do an entire separate hour just on any one of these 4. If this kind of deep dive is your thing, hit that like button. It actually does help push this stuff in front of more people who'd probably enjoy it just as much as you clearly did by making it this far. And if you're not subscribed yet, friends, I don't know what you're waiting for at this point. We do this basically every week, chasing down whatever weird, half-confirmed, gloriously contested corner of science has been keeping me up at night, and bringing it back here to argue about with you. Until next time, stay curious, keep asking the annoying follow-up question, even when everyone else in the room seems satisfied with the simple answer, and I'll see you in the next one.